one small bottle of water.
I would like to introduce him before I hand over the mic to him. So, sir has done his Masters from Tamil Nadu Agriculture University long time ago in 1992, and then with PhD from Calcutta University 1996. And he has done rigorous postdoctoral training from Kansas State University and Indian Institute of Science, Bangalore. He has been awarded several awards, like Fellow of National Academy of Science, India. Also, he has been awarded the IMSA Best Teacher Award in 2022. And for his research, he is extensively working on plant immunity. So, working on the plant molecular biology, plant physiology, plant microbiology. So, today we will be talking about the genetically modified crops, problem and prospects. It's a very relevant topic to the society nowadays. We are debating with conventional food or genetically modified food. So I'll talk about it. So with this, I would like to request sir to take the charge. Thank you, sir.
Thank you so much.
Yes, sir.
Good morning. Vikas, you speak to all of you. And as you have learned just now that I work on plant immune response, very different thing. But this topic I teach in the classes. So it is not that I am unaware of this. Specifically, I chosen this because I have learned that you are very heterogeneous group. You are from geography, from physics, from chemistry and biology, many of them there. So I would not keep any technical component. I'll just speak very thing general about how acceptable the genetically modified crops are and what are the problems associated with them, and are they genuine problem or if we can ignore them. Okay. So I am from School of Life Science and you are here somewhere in this campus only. This is JNU campus, aerial view. Oh, sorry. So the lecture outline that I'll speak about genes. All of us read in our school time, a class at least 10, 12 we have read what are genes. I do not know, many of you might not know what is transgene if you have never read biology after class 12. So we'll speak little about genetic modifications, knowledge resource, where do you get it from, what are the safety concerns you may have on them, and something which I had taken very recently, last about 10 years, is genome editing. So we'll speak about that and then something about natural versus GM crops. Okay. So I'll begin with this topic. Hello, is it better? Okay. I thought this works, it doesn't work then. Anyway, so we'll speak about genes and transgenes. See, definition of genes are very complex. So all of us know, we speak about it is in my gene. I can't do it because it is in my gene, or I can do it because it is my gene. But similar vague concept is there even in the textbook. I mean, gene we have heard, but definition of gene is the toughest thing to speak about. What is gene? We do not, very difficult to define. But simplistically, we can define genes are the ones that define any individual's all characteristics, phenotypic characteristics, behavioral characteristics, morphological, whatever we do, even our thinking process, everything is controlled by genes. And the first concept of genes came from the concept of inheritance. Like, I inherited the genes from my parents. All of you have inherited all of your genes of parents. So we have the resemblance to our parents. Our children look like us. That is inheritance. And that all defined by the genes, and experimentally that was demonstrated in 1884 to be precise by Gregor Johann Mendel. He was from Vienna, and he was also from School of Physics in University of Austria. And then at that time, that was a protocol there. The professors has to be taken charge role, so he was assigned to a charge, and in the charge he did experiments in the garden of the church, and he identified that everything in a plant, or actually he worked on plant, is governed by genes. Like if he has pea plants, some are tall, some are short, and then he crossed them and found that the first cross is tall, and then next generation they segregate out. So he gave the theory of inheritance, and that is the earliest concept of the gene, which regulates everything. Okay. And then, what did I do? I have a physical entity. Like you see, what I told you earlier is the character, inheritance. But the physical entity of gene lies within our cell. Inside the cell you have chromosomes. Like say, this is the cell, you have nucleus, then chromosome. Each chromosome is composed of a stretch of DNA, and this is primarily the physical entity of DNA, and chemically these are nucleotides, and series of nucleotides make it genes. So like as 1884 the inheritance theory was published. It was not accepted until it was reproduced by others. So 1902 it was kind of accepted, theory of inheritance was accepted, and then people started breeding for making better crop plants, better animal, better poultry, started making it. So this is one of the earliest story. In 1917 Stackman is breeding for Puccinia graminis resistance in wheat. So there are some varieties which were resistant, some were susceptible cultivar. So he would try to make the breeding and then get a resistant variety. So if you strictly speak, then you would say this is the first maybe ever transgenic generation, because when you breed two individuals artificially, you actually mix the genes from one to other. Okay, though we do not say transgene, because there is another similar word called cisgene. Cis means within the species, trans means from a different species. Breeding is possible in an interspecies, that means within a species the breeding is possible. So we do not use the word transgene, but definitely gene came from a different plant. And so this was the wheat rust, and one of the earliest success in conferring resistance in wheat. And then similar many things are there. This is one of the example. Yesterday Professor Ramesh Shanti gave a talk. He was talking about the space biotechnology. The same person actually has generated this variety called improved Samba Masuri. This is a resistance to bacterial leaf blight disease. So what he did, he found some wild type rice which was resistant to bacterial leaf blight. Then he made several crosses, and this was the original Samba Masuri, and this is not transformed, bred to a better resistant to bacterial blight. And in a given season, you see this crop field got a lot of bacterial blight naturally, but this is very resistant. So people could transfer the gene by breeding from one individual to other individual. And then there is a mass transfer. See, the Puccinia resistance in the wheat or BLB resistance in rice, they are transferring one gene by breeding many times. But you all have heard about Green Revolution, right? From 1900 around 60 to 1970, this one decade, there was a Green Revolution in India, Bangladesh, Pakistan, Afghanistan, Nepal, Myanmar, Vietnam. So there is a large part of these Asian countries underwent a green revolution when yield suddenly increased more than 3 to 4 times. And that all is done by again transferring genes from one variety to other. Actually, when I was child, if I walk in a not child, I'm talking when in my BSc, MSc, when I was tall enough like me, if I am walking on the rice field, my head will be covered. I mean, I would be invisible in the rice field. But nowadays, any of you who walk, it is up to even not knee height, less than knee height. So the rice plants become shorter, so is the wheat. And those days, specifically in September, October, I remember if any mild wind comes, the all rice will fall down because of the heavy head weight and the tall plants. So there is used frequent famine and all. So the green Dr. Norman Borlaug, what he did actually, he took Japanese dwarf rice and bred with Indian indica rice. Indica rice is tall, and then transferred the dwarfs into indica rice. Similarly, he took dwarf wheat from Mexico and bred into Indian wheat. Indian wheat also became shorter, more resistant to other natural calamities including wind and other things. So these are the primary cause how green revolution worked in India, in addition to irrigation and fertilizer. So in India, Dr. M.S. Swaminathan was his collaborator and who did extensive work in India, who passed away only a few years back. Now, whatever actually I was giving the example, these all required gene introgression from one plant to another plant. Okay. But there is a limit how much you can get from wild. Like you don't get everything from the wild life plant, right? You have the genetic resources are limited, what you can get it. Then people started thinking about getting new and new character. So the new and new character you can get by making mutation. So mutation also people learned as early as 1950s, and then started doing mutation in many crops. So you have mutating agent like gamma ray, X-ray, different chemicals, UV light, they can cause change in the DNA. And then people started developing better crop plants by mutational breeding. So you mutate, you will get random changes, but these are not specific change. Like what happened, a gamma ray can make deletion. A few base pair can be deleted in gamma ray, but you cannot control where the deletion in the take place in the genome. Genome is very big. Where it actually delete you cannot control. It will delete everywhere or many places. Then one has to look and screen for the better plant, then identify where the mutation is. That is possible. So it's all random mutagenesis. So you get random changes, then screen for desirable traits, and that you need to backcross many times because there will be many undesirable mutations. Some will be desirable, many will be undesirable. If you backcross many times to a elite variety, then you can remove the backgrounds. So this is what people have done it, and a good number of varieties have generated by mutational breeding. So because the mutation is done by human intervention, you can tell that is a kind of a change or edited crop. Changed or genome or edited crop, because this is not something not happened naturally. Human have done it. Okay. But one thing is that people have accepted mutated crop in. There are mutated crop varieties in rice, wheat, in brassica, in India. Many of us are cultivating and eating that food. So none of the government anywhere in the world have ever opposed mutational bred crop. Okay, even though they are actually kind of a human intervention and human made. Now you see whether you do a conventional breeding from the wild type plant or do a mutational breeding, this has a big limitation. What is the limitation? That you can have gene transfer only within a species, not in a different species. For example, there are many bacteria which can infect both rice and wheat. If you get a resistance genes from wheat, you cannot transfer to rice because you cannot cross a wheat to a rice. They are two different genus and species. You cannot cross them. So mutational breeding or natural breeding is limited to given species only. It cannot be different species or different genera. You cannot do that. So that's a big limitation. But that eventually overcome by a technology which we popularly call as genetic modification or transgenic plants. So humans have learned from the bacteria. There is a disease called crown gall disease. This is Dhatura plant, and there is a crown gall disease. It looks like cauliflower because of the tumor growing. Tumor was identified long time back, around 1902, 1903. But in 1970s, we learned that tumor is happening because bacteria is transferring a part of its gene into plant genome. So it's a natural genome transformation or genome editing, you can say so, that bacteria transfer their genes into plants. And the first time actually, these are the two faculty members in University of Belgium, Ghana, University of Ghent, Belgium. They mastered the technology that what they did, they took the same bacteria, removed bacterial genes and put different genes. And they found that a foreign gene can be delivered to the plant. So by 1970s, people have learned that you can make a transgenic plant and a foreign DNA can be delivered into a plant through a bacteria. And later on, many different technologies have come up. Transgenics have gone into a kind of a summer training job. People are coming within 15-20 days, they are getting some transformed and going back. So this is the team from the University of Ghent, and this is the first transgenic set of plants, and it was generated by an antibiotic called kanamycin. So if a plant is resistant, because actually the process is that when you select a transgenic plant, you have to have some antibiotic resistance so that non-transgenics don't survive. Okay, so they used kanamycin, and they found that even kanamycin resistance is following Mendelian inheritance. Like Mendelian inheritance is like 3:1 if you have one gene. So they found that there are some of them are dying, some of them are surviving, as you would expect in the Mendelian inheritance. When I told you that tall plants of the F1, next generation segregate both tall and short. So similarly like that only. And then you can see here, this is the first transgenic crop plants. It's a tobacco. What they did, they made it resistant to herbicide. Actually in India and elsewhere in the world, we spend a lot of labor to remove the weeds. In the field you get a lot of weeds. Now what they have done, like they have made this plant transgenic for a weedicide resistance. It's a chemical resistance. Now the same chemical, its name is Basta, if you apply on control or an untransformed plant, the plant will die. But the transgenic plants, they don't die, because transgenic plants have a gene which can make them resistant to herbicide. So that was something, the first batch of plants came up, and in no time almost. So it was 1984 paper was published. 1992, so within 8 years, transgenic soybean, transgenic canola and transgenic corn or maize went into farmers' field within 8 years, because that is only in US and Canada, because US and Canada labor charge is very high, so they cannot afford too much of labor cost to remove the weeds, but chemicals are plenty and easily available. So very soon actually the technology went into the field, and people are generating these transgenic plants in the farmers' fields. Then 1992, actually, you know, all of you probably, I do not know, all of you know about it not. Most of the plants or crops what we grow are hybrid seeds. That means two different species or two different varieties are crossed. The yield is very high, hybrid seeds. In 1992, a technology came, like hybrid seed generations required crossing. You have to cross individually. In 1992, they made a transgenic tool for making hybrid seeds. So what they did, you look here, it's very interesting. This is a transgenic plant where, say in a wild, these are anther and these are gynoecium and these are androecium. Androecium is the male organ and gynoecium is the female organ. So in a normal plant, you have both androecium and gynoecium, and this is a cross section of the anther. You can see the pollen grains. Now the transgenic plant they have done, they have specifically expressed one toxin gene in the pollen grain. You see this plant, everything is good except the pollens are not developed. Pollen grains are not developed. So what happened? This transgenic plant became female. But there must be technology for surviving those plants. So there is another gene called burster. So anyway, this is a toxin, this is inhibitor of toxin. So when toxin plus inhibitor made, that's a common plant that can be used as a source of the pollen donor, and this can be the source of a pollen receiver on the female plant. So you can just now grow the transgenic plants side by side and you can harvest seeds as a hybrid seed. You do not have to do any crossing, no manual crossing is required. So that was the first time developed in 1992. So you can see the speed of science, how it moved. It's pretty fast. The transgenic first batch of canola plants were ready. This is a very nice graph, actually I like it. You see, this is 1996. Actually 1992 was developed, but the farmers field it was in agricultural experimental land for 3-4 years. Then from 1996 it went to the farmer fields. And if you see the year wise, every year almost people are increasing more and more amount of transgenic crops, and these red bars are from the developed countries and these green bars are the developing countries. So the developing countries, specifically Brazil, have come up in a big way to accept transgenic plant for last 7-8 years or 10 years. So you can see the developing countries are lagging behind. So from around 2012, Brazil actually came up almost equivalent to US and Canada. So developing countries are almost now have crossed the developed countries in terms of million of hectares of land used in transgenic plants. But one interesting thing also I would like to tell you here, that I googled it yesterday. Total cultivable land in the world is 1.6 billion hectare, and if you see total, around 210 only hectare is used as a transgenic in worldwide in 2024, which suggests only about 13% of the world land, agricultural land, is being used in transgenic crops. 87% is still being used as a non-transgenic. Global statistics in 2016, it is really very surprising. If you see in entire world global statistics, 90% of canola, 88% of corn, 90% of cotton and 94% of soybean are transgenic. Okay. In India, we don't allow any food crop except cotton. We are allowing cotton, and in India cotton is now 100% transgenic, near 100% transgenic. There is no non-transgenic cotton in India. But other crop plants are not in India. Like India doesn't grow much of soybean or corn or canola. They are Mexico, Argentina, Brazil and US. They are making these crops and mostly they are transgenic. But India is also not importing any transgenic crops. So whatever the Kellogg's and other companies are supplying corn to India, they are supplying only the non-transgenic corn in India. So we are not eating anything. We are very conserved in that sense. There is a website, if you want you can go there. It's called International Service for the Acquisition of Agribiotech Applications. In short, it's called ISAAA. So ISAAA is a website kind of thing which monitors the transgenic plant which is being grown worldwide. Every country, every data they have it. So, and they are almost every 15 days they are publishing a bulletin about the transgenic crop status. And these are the different crop plants which are in the farmers' field across the world. And these are the different countries which have approved transgenic crops in their world, in their crops. So you can see we have India here, we have Pakistan here. Bangladesh actually growing almost more transgenic than India, because they have approved Bt brinjal. Bt is an insect resistance mechanism. Bacillus thuringiensis is a bacteria that can kill insects. So transgenic plant which are called Bt plants, like Bt cotton we have in India, these cotton plants are resistant to insect like bollworm. Similarly, brinjal have a fruit borer. So if you do not spray insecticides, it's very difficult to grow brinjal plant. You get a lot of insects. But transgenic brinjal is insect free, not only insecticide free. It's very important, because if you have a Bt crop, you don't spray insecticide. They are natural insect resistance. So they are insect free, insecticide free. They are very good. Bangladesh has approved. Pakistan has approved Bt corn recently. But still Bt brinjal I'm not sure. But if you want, you can check, like in this website you can click here in the Pakistan and see what are the transgenic plants they are growing. Everything you will come to know. And these are different traits which are being approved: abiotic stress, growth, yield, disease resistance, herbicide resistance, quality, quantity, pollination control. Many things are pollination control means I told you about Barnage hybrid seed production. Okay. So this is a data, kind of a latest data published by Agro Investor 2025 that gave you the different countrywide how much transgenic crops are being grown in their field. And if you see here, the highest is United States. That is what I'm telling you, followed by Brazil. Brazil started much late, 2000 almost 10 or 11 they started. But actually they have widely accepted almost every transgenic plant by now. And they are hugely growing soybean and cotton and corn. So Brazil is the second, Argentina, Canada, India, Paraguay, China, South Africa, Pakistan, Bolivia and others. So these are the major places who are growing transgenic crops in their fields. And this is quite also again surprising. If you see the crops, you see only 4-5 crops are majorly grown in the field. There are many others in the pipeline or just started. But you know, this counting, I have read the paper, they have not counted genome edited crops. One is transgenic, one is genome edited. I'll speak about it. Genome edited banana is recently has been nodded in Africa. So Africa very soon is going to adopt genome edited banana plant. And India may approve genome edited rice, genome edited brinjal. So that is also pipeline. Maybe another 4-5 years it will come to the field. So situation may completely change. But right now, soybean, maize, cotton, rapeseed, sugar beet and sugarcane. These are the major plants which are transgenic and being grown in the farmer's field. So this is the last data slide about the statistical data. I wanted to show you here because look at the percent. This is the amount of cotton grown in India from 2002 to 2016. India adopted Bt cotton in 2002, merely began with very little amount of land in Maharashtra. Then Maharashtra, Gujarat, MP, Chhattisgarh, everywhere now cotton is actually being grown almost near 100%. Look here, in 2016, 100%, near 100% cottons are transgenic. So that gives you one idea that, like many times we debate whether transgenics are good or bad. You see, farmers are not fool. They are not foolish. They know very well which one is going to give them better food, better output, right? So when farmers have adopted 100% land area for cotton, and the only crop which is allowed in India, you can easily assume return is better. Return is better than the non-transgenic or conventional one. There are many people who oppose GM. I will speak about that. But this kind of statistics definitely tells you that there is something in the transgenic crop, and farmers are definitely getting more benefit. Otherwise, they would have not accepted. Right? Now, tell me if you have any question from this part. See, this is same as the conventional varieties also. What people are doing, like now the cotton, there are at least 4 or 5 different varieties are available. So one variety like rice, for example, is grown from Kanyakumari to Kashmir. Rice is grown, and different varieties are there. You cannot have one variety throughout the country, right? So if genetically modified rice is allowed in India, you will find that it is coming in 20 different varieties at least. So that according to your agroclimatic situation, you need to adopt that variety. Now, if the natural calamities affect, it will affect both. That is a different thing, right? It's insect resistant. It is not a natural calamity resistant. So it will be similarly affected both.
Suppose there is a bacterial gene inside that cotton. I think there is a mini cotton.
Right, insect resistant gene.
So this is against the pesticide. In the climatic conditions changing, the bacterial gene is performing the same manner or essentially.
Okay, actually that is a continuous process. So if bacteria changes their mode or anything, then people have to change the gene also. This is a continuous process. It cannot be done once and it is done. Right.
India, what criteria some crop is allowed to go for the transgenic crop? Suppose BT cotton is allowed.
No, India has not allowed any food crop. Among the crops you can see soybean, corn, sugarcane and sugar beet. That's are the ones. But the only cotton is the non-food crop. India has allowed non-food crop. So that is why only cotton so far.
And that's why BT brinjal are not allowed.
Not allowed, right. Okay, so any other question? Yeah, please.
So, the nutritional value of the genetic modified crops and the indigenous crops, so are they different?
Nutritional values, see, they are thoroughly analyzed. They are thoroughly analyzed, lot of data available. Should not be different. Should not be different. If you are putting a BT cotton gene, BT gene, it should not be different from the normal one, conventional one. Yes, please. Variance? Which they started. Now they are putting at least 4 different cry genes. So cry1 AC was mainly for the lepidopteran larva. Now you have a coleopteran, you have hemipteran insects, other insects are there. So now people are putting many different E gene, many different gene pyramiding, so that not one bacteria, one insect, at least 10 different insects can be controlled. So those are in pipeline. But what we have to do, we have to open up for the technology to be accepted, and then maybe more and more gene can be put in. right? So I will go to that. I mean, you can again take the question later, no problem. So now the question is that something which we have started discussing from your question and her question is that whether really GM crops are good or bad. So this is a debate started in 2002 onwards. It's still continuing, and we'll speak something about it which could have been taken now. One thing is probably many of us are aware that frequently there are protests many places in the country, in Delhi, Bangalore, that there are groups who are protesting against GM crop. So there are anti-GM group. If you Google one name called Bandana Shiva, she is from Bangalore, from a company. She started a movement for anti-GM movement, and there are many others. And even this is the protesters in Norfolk and UK. They are vandalizing the GM field. So there are protesting groups who are fiercely protesting against GM crop worldwide, not only India, almost everywhere in the world. So they are highly afraid that GM crops are going to be harmful for the society, for the nature, for the atmosphere, microbiota, everything. So everything they are scared of. So we'll speak something about it, that what are the really issues they talk about. Okay. So what I have written is what anti-GM groups say. Okay. Please note, this is not my saying. Huh? I am writing. I am telling you. I'll tell you both the components. What anti-GM people say and what are their counter argument by the GM supporting group. Okay, keep me as a neutral, as an informer only. Presence of antibiotic resistance gene. So very early slide of the Jeff Shell and Bhavan Mantagu's lab, I showed you that you need to have a kanamycin resistance gene for selecting transgenic, right? So their main one of the concern is that you have an antibiotic resistance gene in the transgenic. So that's bad, because your antibiotic resistance genes eventually can go to animal, can go to microbe, and can really make catastrophe. Now counter argument is that most of the varieties which are released to the field, antibiotic resistance gene has been removed. There are technology, so biologists definitely would know. There is something called Cre-Lox technology. So there is a technology you can remove the part of the gene which you don't want. And in fact, the new concept is zero tolerance. Zero tolerance means you have a gene you want to put, add that. No base pair extra can be added. So majority of the varieties which are being released in any other countries on the food crop, they have a zero tolerance. Means no extra bases. Suppose you want to give a gene X, have gene X only, nothing else. So that is being made now. Antibiotic resistant genes are removed. So, but still they say that it could have unintended effect which you do not know. So the unintended consequences. They say that human civilization is more than 10,000 years old. Last 10,000 years we are eating natural food, and now suddenly we started making artificial food. We do not know how this is going to affect. Should you really accept it, which is just started in 20 years ago or 15 years ago. So this is their logic, that we do not know what could be the consequence of it. Another thing is the pollens through the wind could pollinate wild varieties of the modified plant, producing hybrid with strange characteristics. This is, plants have the pollens can fly and they can contaminate to the nearby field. So you cannot restrict them. It is not possible to separate these crops from other varieties. That's the same thing over here. The risk of these crops interfering the environment, these risks have not been thoroughly evaluated. So these are the kind of major arguments which are given by the anti-GM, which they don't like it, and very vocal and protesting worldwide. The something we'll just discuss a little bit, that lack of adequate testing of the GM crop. Now there is a catch. Catch is, if you look into ESDA, United States Department of Agriculture data, which are major grower of the transgenic crop plants, they give in their website all data for the field study and safety study. US specifically is probably 100 times more concerned about the health and safety than Indians and other countries. One example is that you cannot even buy antibiotic medicine or prescribed medicine from the shop. If you go to any construction site, you will not find single rod is that we have houses are constructed, every rods are open, and the trucks are carrying rods and bikes are pushing in the back and getting killed. In US, you will never find single truck carrying open rods. I have seen a construction building in the Kansas State University. Every rod they're putting a fluorescent glow red fluorescence on the rod. I was so surprised that even in the night from the plane, they will be able to see there are rods in the down. Every rod has a fluorescent marker. They're so careful, concerned about everything. They cannot accept so much of transgenic crop without thoroughly testing. Each and every crop that is being taken as a food in US are thoroughly tested. So it is not that correct. But the catch is, what the anti-GM people are saying, who is doing the testing? In India, BT cotton is being sold by a company called Monsanto, and Monsanto has collaborated with a company called Myco, Maharashtra Hybrid Seed Company, Myco. Myco is selling the Monsanto seeds to BT cotton in India. Who has done the field test? ICAR did not do it. It has been done by the Myco in their field trials. Anti-GM crop group has a very valid logic that did USDA themselves tried in their tests? No, they relied the data which is generated by the private company like AgroDo, Myco, Pioneer, Monsanto. These are the companies which are making hybrid seed. They are testing, and they are only observer from USDA. USDA observers are going and checking at them. So they say it is not being tested openly by the publicly supported institutions. In India, ICAR did not do any trial. Trial has been made by the Myco in their seed factory. So there is a big fallacy in the way of testing that is genuinely correct. So anti-GM groups are not absolutely wrong what they said. Testing is not done by the public. See, ICAR, Indian Council of Agriculture and Research, they have field in every district of India. Suppose BT brinjal would be allowed today, why it should not go to the ICAR field? Why it should go to the seed company field and being tested? That is a genuine concept. So anti-GM groups are saying that in countries like US, GM food crops contain ingredients level. Okay, another concept. Like in US or other developed countries, if you go to supermarket, you have one corner for the GM crops. There is a label is there in each and every crop that this is GM. If you want to buy, you buy. If you don't like to eat a GM, you don't eat them. But India kind of country, that system will not collapse. Thelawala will come with the sabji and lelo, vegan lelo. They will never tell you that this is transgenic, this is non-transgenic. Aap kaun sa lena hai bhai? So actually they say that in developing countries, giving a transgenic food will make a complete chaos. People will not know what they are eating, and what they say, the right to the knowledge, right to the information. As a consumer, I must know what I am eating. If I am eating a GM crop, I should know that I am eating a GM crop. That is the anti-GM group are saying that in developing countries you cannot control. There is another big concern, is the patent and or monopoly. You see, many of the cases people have linked in Maharashtra, farmers suicide to cotton failure, and many of them say that they purchased cotton seeds at a very high rate. The seed companies don't allow the seeds to be collected. Actually, you can Google it, you will find out. In Canada, a seed company, Monsanto, sued a farmer for $5 million because he grew his own transgenic seeds on the field. So you can Google it, you found it. Then after 5 years, it went to the court. Court found the farmer as guilty, but waived off the fine. They said, okay, as a farmer, you are serving the society, you don't have to pay the fine, but you are guilty, don't do it again. So, the seed companies which are making seeds, transgenic seeds, selling them into the farmer, they took patent of that and monopoly. Not only that, farmers signed the agreement that they will not grow their own seeds, they will buy again in next year. So what anti-GM said, that as a farmer, farmers should have right to collect their own seeds, right? Now seed companies like Monsanto and Myco and everything, they know that they cannot control farmer of India and other things, because Indian farmers will do it anyway. So what they have done, they put hybrid seed. Hybrid seed is a technology I told you earlier. You can cross two varieties, get a very good quality. So if you buy seed from Monsanto or Myco, your cotton yield will be very high. But if you collect from their seed yourself, you will find it will be very, some part of the field will have a good yield, some part of the field will have a very bad yield. Because hybrid seeds segregate in the next year, next generation. So this is the technology what seed companies are doing in developing countries. They are all selling only hybrid seed. That's a genuine problem. Question is that there are simple solutions, but I do not know why governments are not doing it. The simple solution was ICAR could have been, taken the lead. ICAR could have generated the one is called hybrid seeds and one is called true breeding seeds. There are many varieties, classical varieties are true breeding. You harvest seed, it will be exactly like as it was the previous generation. So why the government cannot step in, cannot give truly breeding good quality seed to the farmer? No idea. No idea what the governments are doing, not only the present government, past governments. None of the governments have come forward to say that common will do it. We'll do it for the farmers. We'll give them the good quality seeds, and it should be very insect resistance and true breeding. Never happened in any country, including India. Now again I am going back to the previous question, like say, if GM crops were allowed, so if GM crops are not patented, farmers are given free GMO with the right to save their own seeds, will that be accepted? So see, the arguments are they have a monopoly, they have antibiotic resistance and many things, right? So the counter argument I have put here, suppose there was nothing like that, it was a true breeding, it was very good, will the anti-GM people shut down and take it accepted easily? No. Golden rice is a golden example. Golden rice was taken to take care of the vitamin A deficiency. Vitamin A is a fat soluble vitamin and almost absent in rice. You can get in fresh vegetables, you can get in meat, fish, oil, but you don't get anything in cereal crops like rice, wheat. There is almost nothing. Now specifically the problem in the African countries, some of the country people get child after above 14 they become blind. Some countries actually everybody is blind after 14 because of the vitamin A deficiency. So specifically in African countries which eat only rice and they don't have the luxury to eat vegetables and other crops, and so a child get blind after a few years. So this is gentleman called Ingo Potricus. This Ingo Potricus is a professor in Switzerland, and his laboratory, 10 PhD students almost worked for 10 years, and he developed something called golden rice. So he was in the cover page of the Times magazine on that 2010. The paper was published in 2009 or 2000. Oh no, Science 2000. Ingo Potricus made true bidding golden rice, which will not follow up in the next generation, did not take a patent, and took initiative through different NGOs to give them free rice seeds in the African countries, okay, which are making vitamin A. You see here, this is a just brief science about vitamin A. This is actually starts from in IPP, inositol pyrophosphate, and then there are many steps. And if you see, rice doesn't have these 4 genes. What Ingo Potricus did, they got one daffodil gene, one bacterial gene, and another 2 daffodil genes, one bacterial genes. They made in rice. Now rice can make vitamin D, vitamin A precursor. It is actually become real golden color because vitamin A is a carotenoid. Carotenoid has a golden color. So the rice became golden color rice. So they gave a golden rice. Look at the carotenoid purified from the wild type, nothing. These are the peaks in the HPLC showing carotenoid peaks. So transgenic rice was having a good amount of carotenoids. Was the golden rice accepted by anti-GM groups? No, they did not. What the arguments I said? A little vitamin A. Actually, vitamin A had 1.6 milligram per kg of rice. The transgenic golden rice had 1.6 milligram per kg, and daily requirement is 0.7 milligram. That means one has to eat 700 to 800 gram of rice per day. They said, oh my God, who will eat 800 gram of rice per day? So better no. And then antibiotic marker genes, that was actually removed, and a 35 is viral promoter. Now look at the logic. You don't get anything versus you are getting some amount, good amount. Actually, if people get 200 gram of rice, it should be at least 1/3 of vitamin A they should get it. But it was fiercely protested. Okay, come on. Then something came called golden rice 2, like our mobile 2G, 4G, 5G. Golden rice 2 came in 2005. Paper was published in Nature Biotechnology. Look what they did. 35 microgram of carotenoid instead of 1.6 microgram. That means now they have come to a stage that if you eat 150 gram of rice per day, you should get enough vitamin A. What they did, they removed viral promoter, they gave rice promoter, and they did not use any antibiotic resistance gene also. They are the sugar selection. They are called sugar based selection. So golden rice 2 was made with addressing all the queries of the anti-GM group. You have enough vitamin A, there is no antibiotic resistance, there is no viral promoter, there is no question of anything which is coming should be bad for human consumption. Did they accept it? And another thing is the golden rice has never got patented. Ingo Potricus was very firm enough that it should go to the human being without patented. No chemical application involved. Highly beneficial for large part of world. This is from the article journal 2018 biochemical journal. Look at the written. Golden rice enriched in provitamin A by the humanitarian golden rice project. Despite its humanitarian and non-commercial nature, golden rice is fiercely opposed by anti-GMO group. They did not accept. Even for the anti-GM group protested much more. And they said because it may potentially lead to a better public acceptance of other GMOs. They said, अरे golden rice खा लिया तो बाकी transgenic भी खा लेंगे, क्या फर्क पड़ता है? And with that apprehension, people have fiercely opposed golden rice too. So this is what the situation. One has to look at see the balance between benefit versus potential harm. when millions of people are getting blind because of vitamin A, people need to learn to accept it. But there is some ray of hope. Africa very recently has allowed transgenic plants in their field. So maybe in another 6-7 years, you will see the golden rice too is being really grown in African fields. Very recently they have approved. African governments are no less than Indian governments. Both government-wise, actually almost every country is similar. Okay, this is a pretty brinjal story of India. So it still has not. Bangladesh have started now. Bangladesh almost 70-80% brinjal is BT brinjal, but India has not accepted. BT brinjal could give the fruit borer, and so this 2002, 2009 it was almost going to the field of Maharashtra farm field for field trial. Forget about farmers field. Even field trials are not being allowed. Jairam Ramesh was the, I think, agriculture minister in Congress government before BJP came up. So that time lot of activists used to say that Congress government never took any decision. They are decisionless government. They could not make any decision. Especially BJP was anti-Congress for the lack of decision. They have always blamed Kashmir issues and many other things that Congress are decisionless. They cannot make decisions. So they said, actually many of them say, if we come into the power, we will take decisions. But ultimately the vote bank that matters for any political parties. You see, from last 10 years, 12 years, BJP is in our government. Do you see BT brinjal in the field? Field trial, not even field I am talking about, field. No, not yet. Because less number of scientists give the vote than the people who are not giving vote, who are anti-GM. So none of the government has the audacity to challenge the vote bank, and that is fact. So same thing, I am not telling about BJP specifically or Congress specifically or any country in the Africa or anywhere. People, any democratic country are afraid of the vote bank, and they would mainly not take a step without calculating how much is going to affect the vote bank. So BT brinjal story is now in the cold storage from 2002 onwards. From 2009 it was supposed to come, and then again went to the cold storage now. As of now it is in cold storage. Okay, fine, forget it. Good. So now is the last component almost, the genome editing. If you have any question, you can ask me now. No, but who will give the seeds? Seeds actually are not allowed now. However, they will get the seed from? No. Actually, government has to allow first. And before that, safety test must be done. Field trial has been not done. Forget about, this has been done only in the laboratories. Inside the laboratory, in the greenhouse, in the transgenic facility, people have tested. So before going to the farmer field, at least 4 or 5 generations it will be tested in the what you called as a field, which are monitored and isolated. Isolated field it will be tested first. Then if it is good, then it will be transferred to the farmer's field. So it is still not come to the field trial situation. So it is something like clinical trial for the medicines. So clinical trial has not taken place. Forget about the application to the human. Okay. So it has stopped at the clinical trial level. That we do not know. What is happening in our Chabis Pargana and other places, what is happening is we do not know. Maybe some amount of BT brinjal is going there, I do not know. Well, at least not officially. Okay.
Ultimately, it's under consideration in history of environment, right?
Right.
So it's a concern of the environmental
Actually, there are many steps. GESC is the highest governing body which has to approve. They take the consent of all other ministries. So essentially, ministries have not given clearance, and not because of the assay, primarily because of the vote bank politics. Because you see, unless you do the test, how will know how it's affecting or not, right?
It is under domain of Ministry of Environment Forest Direction.
Actually, it's headed by the GESC, Genetic Engineering Accreditation Committee. That is the one which is highest body. All other ministers are under it. Okay, so everybody is involved. But there is a ray of hope. What is the ray of hope? It's genome editing. Genome editing, but one thing I'll tell you, I would like to say you something also. You look here. Look here at the plateau. Look at this is the red color is the developed countries. Developed countries have come to a plateau. So, if everything is good about genetically modified crop, then why this is not growing any further? Why from 2016, 15, 16 is a plateau now? That's also something to think about it. So maybe even the developed countries have started thinking that it's not all good. Because technology is there, any other crop plants could have come. Of course, there is another counter logic, is the land area in developed countries is limited compared to the developing countries. Developing countries have larger land areas. So, developing countries are still increasing. That is possible. But look, this is only till... Huh? No, we are talking about the land area coverage. Now you see, like Europe, US, Canada, they do not need, because they have abundant of land. So they do not need, actually, if you see about the area, yield per hectare, we need to increase a lot. Because our population is more, our land area is much less per human being. Australia doesn't need it. Europe's don't need it. But we need it. China needs it. India needs it. Bangladesh, Pakistan, Afghanistan, they need it. Our land area is much less per capita.
So one more question. Is the two crops or the transgenic crop or is the phenotype also?
No, phenotypes are often better. Phenotypes are better.
Two crops here in the front of me, I can't identify that
No, you cannot identify. So a good thing is actually that almost every country have started adopting and approving GM crops. We mean the genome edited crops. That's also a GM crop. But the difference, actually I'll try to explain what is the difference. See, what happens in the transgenic, you are putting a foreign gene in a plant or animal. Now, earlier people did mutational breeding. During mutational breeding, you have randomly changed the genome here and there, anywhere, then select the better one. But the genome editing tool allows you to change DNA in a given species sequence. So it is like a mutational breeding only, but you cannot do this mutation without giving it the transgenic. So there is a catch. Catch is you have to transiently make it transgenic and then make the genome edited by remove the transgene. Okay, but new technologies are coming up. Only last week there is a PNS paper. PNS paper, they have shown you can make transgenic by virus, a genome editing by virus. So any of you are interested, I'll give you my email ID. You can write to me. I can send you this paper from PNS that was published last week, not last week, 2 weeks back. You can use TRV and some viral vector to make transgenic genome editing without making transgenic. So that is also technologies are coming up. But the good thing is that European countries and India have opened up for the genome edited crops. That is a new hope. Now, what is genome editing? As I told you, that it's a kind of a precise mutation. Suppose you have a DNA, anywhere in the DNA means actually genes, lines of genes are DNA. You make a cut. If you cut, the cell will try to repair. When it repairs, you can make a mutation, because sometimes repair are error prone. You can make a mutation during repair, or you can change by giving a donor DNA which is changed. Actually, this concept is now very close. It's actually almost third phase of clinical trial is complete for gene therapy for blood diseases. I mean, human diseases now are almost in the third phase. Very soon you will find many of the genetic disorder can be cured by genome editing. Suppose if I have a bad gene, if a child has a bad gene, that can be rectified by stem cells in vitro. These stem cells can be injected in the body, and then stem cells can rectify this disease. Thalassemia, and then leukemia, sickle cell anemia. These kind of diseases are almost in the third stage now, and it will be very soon, in a few years, people will do gene therapy thanks to the genome editing technology. This technology is not very old. It's only 15 years old technology, but has really come up into a big field. You make a cut in a DNA and give a donor template. Donor is a corrected genome. So the donor can get into the repair process, and you can change the genome. Any genetic disorder can be corrected that way. People knew it for years, more than decades people knew it. But people did not know how to make a cut in a DNA in a place you want to cut it. So that was the main bottleneck of the genome editing. Otherwise people knew this from more than 30 years, that you can change while repairing, you can correct it. But the people did not know how to make the damage in a precise area. So that came up recently. So, like for example, I'm just giving you the random sequence. In rice you have 430 million bases. In human you have a 3.2 gigabases. In there, if you want any 4 nucleotide, 2 nucleotide, or 1 nucleotide to be changed, you can do it if you can cut here. So wherever you can cut, you can change it. So the earlier one, before 2013, people have done many technologies. So there was a technology people call zinc finger technology. Zinc finger technology was by a nucleus called FOC1 that could cut a DNA. Just briefly tell about it. There was a scientist, Indian scientist in John Hopkins, Srinivasan Chandrasekharan. He found that FOC1 has 2 domains. One domain can bind DNA, one domain can cut. And then what he did, actually, he replaced the DNA binding domain with a transcription factor. The transcription factors can bind DNA sequence precisely in a given sequence. So you can modify a zinc finger, is a DNA binding domain, and a nucleus, you can join together and you can give a cut in a place you want to cut it. So that technology was available from 1992 onwards. But problem was engineering the zinc finger. It was tough, but still people are doing it. Actually, the stem therapy which has been approved now and going into clinical trial has the zinc finger technology, not the new technology. The old technology is being still used for many different reasons. But I don't want to tell, because the group is so heterogeneous, it will be really going out of the limit. But this is a new technology, CRISPR-Cas technology, that allows a cutting the DNA anywhere you want in the genome very precisely, and then anywhere you want to modify it. This technology was invented in 2013, and this is a story. This is the 2013 it came up, so let us forget about it. The full name is there: clustered regularly interspaced short palindromic repeats. This is a bacterial immunity system. I'll skip those jargons of technical thing, and ladies should be happy on this ground that this has been invented by two ladies, and they got Nobel Prize in 2020 for their work for identifying genome editing, which actually reshaped the entire research paradigm in transgenic or gene therapy research worldwide. So, and they got Nobel Prize in chemistry for gene therapy. One is Emurella Charpentier from Berlin, Germany, and one is Jennifer from USA, University of California, Berkeley. Now, what is the technology? Technology is that it was again learned from the viral resistance in bacteria. So, when bacteria get virus infection, bacteria takes up a small part of the genome and put in their resistance locus in bacteria, and then that is called CRISPR. And what they do, when virus comes, they can kill the virus. So one part of the viral genome is integrated, that makes a RNA. This RNA includes a nucleus. RNA binds to the nucleus, and when a new virus comes, their DNA get cleaved by the this earlier viruses. Earlier viruses DNA is incorporated by the bacterial genome, that is used to kill the new virus. So that's what people learned from the bacteria and virus, and now it is being used in the human research. So maybe I'll skip about these things, that's okay. But essentially you can have a RNA molecule. This RNA can bind with the nucleus, and this RNA can guide in the genome. It's called guide RNA. So guide RNA is one single RNA that can bind with one part can bind in the genome, one part can bind with the nucleus. So this is how this is so specific, because RNA DNA binding could be specific. You can make it very specific binding, and you can have a nucleus. Now you can cleave wherever you want to cleave it. So this is a technology which was identified recently and got Nobel Prize. So this is a ray of hope that in 2024 May, very recently, this came in Nature India, that India is trialing gene edited rice after regulatory changes. So GEAC has approved genome edited rice field trial in India. See, Indian scientists from last 30 years are making transgenic plants. There are hundreds of transgenic plants have been generated in Indian laboratories, and many of the faculty members like Professor Deepak Pental retired broken heart that his transgenic plants could never come to the field. He had many transgenic plants that had a wonderful potential. But this is the ray of hope for the new generation scientists. So even if the India is not allowing, we are allowing genome edited. And genome editing has a huge potentiality. You can make marvelous set of crop plants or animal industry farms, poultry farms, which could be genome edited, and that could be used in the field. So very last, I'll take only 5-6 minutes more to discuss about natural food versus GM food. See, this is a slide which I got from transgenic tools in plant cell, and I like the concept, like this one. The concept of natural food is a myth. Means what it says, that there is nothing called natural food. Natural food, like say this one, today is a corn plant. This corn, the human history of agriculture is around 10,000 years old. Okay, when the people started domestication. Before 10,000 years, people never cultivated into nature, did hunting, whatever the crops they got in field they have taken. But about 10,000 years back, people started cultivating their own food. And what they have done, initially this was the teosinte, and now this is the modern corn. This is the coin. This is the size of a coin only. Now this is the coin and this is the cob. So over the thousand of years, people have bred it, selected, and that way the natural teosinte, if you say about, with the human intervention, it has come like this. So it did not come naturally. It has come to this stage by the human intervention. Human intervention has taken everywhere. Look here. This is the early wheat, and this is the modern wheat. Modern wheat is the naked wheat, which doesn't have much of husks. Early wheats have a long on very thick hulls, very tiny grains, and the modern wheat are kind of naked grains with very little on and very thick and big size of wheat grain. That did not happen naturally, all done by human intervention. 7,000 years ago to 500 years ago. By 500 years ago, people have already made it big crops. This is happening in Mexico. Okay. Now you see what we have done. We have removed mountains for mining. This is a picture of a mountain range in coal mine in Boone County in West Virginia. And this is what is the 2009 satellite picture. We have removed entire mountain. We have made big dams worldwide. In fact, China has made such a big dams that tilted the earth by almost a degree. You know that probably. Big dams have removed the earth axis so much. And we have done many places everywhere. So this is 1987 picture, this is 2006 picture. The flow of the river got changed. And there are so many animals got extinct. And majorly because of the human intervention, global warming and everything. Human have bred, these are all artificially bred by human. You have dogs from different sizes. Okay. And you have carrots of all different sizes by breeding. This is a cauliflower which is purple color. It's available in Bengal actually. You can go to, in winter, some parts of Bengal you can buy these kind of cauliflowers. So breeding have made colors of carrots, colored full of combs, and many different varieties of cauliflower. None of them are natural. All of them have been done by human intervention. So the concept what we are saying is the natural food is just a mythological concept. Nothing is natural what we are eating today. A few days back you might have seen in the paper that 1,500 kilometers away people did the robotic surgery. Heart surgery is being done by robots being controlled by 1,000 kilometers away. Right? See what the technology effect. Technologies from the human civilization, we can go by car, we can fly, we can go to space, moon, Mars. You have read about space biotechnology. Robots can clean our home. I have actually robot in my home for kitchen because we are cleaning our home by a robot now. I mean, it's a mini cleaner, they call vacuum cleaner. And robots are surgery. Now tell why agriculture should not use the technology. It's a technology only. Gene transfer is happened earlier by crossings, by breeding. So what is the problem if we use a technology in case of agriculture? Why it should be banned? Modern GM technology offers Technology can now target movement of gene from source to other. In situ genome editing, CRISPR-Cas, marker-free transgenic, I told you zero tolerance, zero extra DNA, precise control of expression, elaborate testing for safety of human, domestic animal and environment. So there should be no problem at all if you think logically that what the GM is doing nowadays are have a very precise transfer. There should be no problem at all. Thorough risk environment is done. I told you earlier, US are very picky about their choices. They would have not allowed GM soybean, their milk. Actually, in US, almost 30% babies take soya milk, because many of them are naturally intolerant, called lactogen intolerance. So many of the 30% of the almost US babies take soybean, and in US 100% soybean is transgenic. So how US is allowing their babies to take soya milk, for example? So it's definitely not unsafe. And from 1996 to 2026, that means 30 years. Actually, if US can survive with their kids starting their soybean milk, transgenic soya milk, then you should not doubt that it could be bad. So this is my last slide. Are GM crops safe to eat? Yes, I strongly believe that yes. This is not my slide, but I support it. That I individually think that GM should be very safe to eat. They are thoroughly tested and less insecticide, and there is no concern should be there that taking GM crop. Thank you so much. Tell me if you have any. Oh my God, thank you. It's okay. It was 2007. They have modified frequently. Now the latest one is 2022. You can download from the DBT website the handbook of handling GM individuals, GM crops. It is available in DBT website. You can do DBT. Otherwise, if you email me, I can send you. My email ID is aknandi. No problem. If you actually, I have downloaded that guideline. I can email you. One minute. There is a question in the back. I'll take it and then. Oh, 100% stable. You know, this is the gene are inherited. Like we have there is a 100% true breeding. If you have homozygous, that means, see, we all have the two chromosomes. So one is coming from the father, one is coming from mother. Now, if you have the same DNA in father and mother, there is not going to change anymore. So before you do after you do the genome editing, you make sure both the chromosome has the same change. Then it is stable for generations of generations. See, there is a new mutation, right? New mutation can happen. Chances, as you have chance in conventional plant, same chance is there in the genome edited plants. No change. Yeah, 1 minute, I'll take it.
So one more thing, recently I studied an article where they are saying that the genome edited or GM modified crops, they are giving very good yield, and the reason behind that, they are extracting more and more nutrients from the soil. So the soil fertility will be reduced quickly.
And that is not logical. That is not logical.
Indigenous crops, they are not extracting.
No. That is not logical. Logical writing. I don't know who has written that. It cannot be logical. So suppose you make a genome edited more insect resistance, why it should take more nutrient from the ground?
To give better yield.
No. That actually, see, anyway, if you have to add more yield, then soil has to give it. Now, see, I don't know if you are from plant background or not.
No, I don't think so.
Okay. In plant, 4% of the nutrient come from the soil, 96 come from the air and water. So plants grow, take very little from the soil, only 4%. 96 come from the air and water. Because carbon, oxygen is the total 90% of the mass, body mass. 6% come from the water, only 4% from the soil. So soil contribution is very little. But still, not negligible, because you get nitrogen, phosphorus, all metals are there. But there are fertilizers. People can add fertilizers. It's not a problem. So that writing is wrong. Wherever you got it, that is not correct. You have a question?
Then from this statement, then can we say that air pollution is a bigger problem?
Which one?
Air pollution is a critical for agriculture.
Air pollution.
Yes.
It's a very different thinking for me.
If you are growing any plant in a polluted environment, then what will happen?
See, air pollution is definitely a bad thing for every individual. It cannot be. Plants, I don't know how much they may be affected because we have a respiratory system. Plants don't have a respiratory system. But see, the things which is bad for us, like carbon monoxide or say diesel particles, which are nanoparticles, as it is affecting human body, it can affect plants also. But they're taken by the stomata. So it's going to affect equally bad for all individuals, including plants.
The second question is this insect resistant varieties. So how they are insect resistant? And what is the mechanism?
Okay, I'll tell you. It's the main mechanism is like we get bacterial disease. Many times bacterial disease can be fatal. We can be killed if we don't take antibiotic or proper treatment. Similarly, insects also get bacterial disease. Insects actually, one of the bacteria called Bacillus thuringiensis. This Bacillus can cause disease in bacteria, they can kill them. Actually, it is equivalent to cholera disease of human. You know cholera disease, how it happens. Actually, cholera makes a toxin. This toxin binds in our gut in the epithelial layer, and that affects the water transport system. So it damages it. All water gets we get dehydrated. So this bacteria Bacillus now binds in the insect gut and can make the gut membrane damage. Insects die. Now what scientists have done it, the bacterial toxin gene has been put in the plant. So if insect eat, the bacterial toxin they will be eating. But these bacteria are very specific. None of the bacteria can affect very specifically. Like pollination is happening by the different bacteria, different insect. Damage is causing by the different insect. So you can have the toxin specific to damaging insect. So your pollinating good insects will not be killed. In contrast, if you spray insecticide, good, bad, everything is going to be killed. So that way transgenic plants are much better because it is killing specific insect, not all insects. Much better than the insecticide.
And that toxin has zero effect on humans.
See, zero effect is very difficult to say. Scientists would never say zero effect. Minimal effect. Yeah.
But this gene product will not go in the fruiting body.
No, not only that. People have done thorough studying. Actually, we don't have the receptor for the Bt toxin. These are the receptor ligand binding system, like that cholera toxin. Cholera toxin bind very specifically, right? So similarly, this kind of toxin has a receptor. Even Lepidoptera and receptor. Lepidoptera is a group of insect, is absent in Hemiptera insects. So it cannot even kill the flies. It can kill the beetles. The ones toxin can kill beetles, cannot kill flies. Forget about human. We don't have in the brass that don't have in the receptors. So chance is very, very low.
So
Yes.
fortification, like you said, for these fortification. So like we are given fortifying biscuits or say toothpaste.
Correct.
It is not necessarily fortification will need absorption. So that kind of study also needs to be done where like you have fortified the plant, but was it absorbed in the human? Was it metabolized? That kind of study also needs to be done.
Right. But what they have done, if you are talking about golden rice, what they have done, what you have in carrot. Say carrot is rich in vitamin A. What you get in carrot is put in the rice. So if human body can absorb carotene from carrot, why can't we absorb from the rice? Actually, that has been tested also. It is actually beneficial. That has been tested, but logically it is the same molecule, right?
Sometimes the absorbable form
That is true. That is true. It may not exactly do as it is doing in the carrot. That is correct. Yes, please.
As I understood from the lecture, it's related to the hybrid process. So, as I know, practically, because I am from our country, so we are planting rice crops, hybrid crops.
Right.
And the treatment, pesticides, fertilizers, release, irrigation, a little bit different from the other crops which is not hybrid. Is there any change in productivity based on the changing of the release?
Most of the cases, their productivity is high. Otherwise, seed company, farmer will not buy from the seed company.
Yeah.
Their yield is anyway high.
Yeah, it is healthy, but-
Otherwise, people will not buy. Farmers will not buy.
Yeah. So the productivity that I am talking about, there is any changes due to changing relief, landscape.
Possible. That is possible. Actually, one thing is correct. Nothing can be achieved without energy. So insect resistance will definitely need a metabolic energy, right? So compared to non-insect resistant, is definitely some metabolic energy will go to make the insect resistance. So yield may be little bit compromised, but you see, you are saving the insecticides, you are saving the atmosphere. People are spraying tons of chemicals like every year. If you go to Bengal, Punjab, anywhere, you see so much of insecticide sprayed. In Punjab, one district is called as a cancer district. Because of the so much of insecticide sprayed, there everybody is having cancer in the family. Same thing is turning into Bengal now. Bengal villagers are getting too much of cancer now because of the spraying of insecticides to the rice fields. So these are very bad compared to what you can good by insect resistant, genetically resistant.
Another thing is, as you know, the amount of insects, types of insects, and distribution of the bacteria, and actually changing geographically based on the
Ecological changes.
So then it will be back to the same.
Now I'll tell you one thing. If you are spraying, how much ecological impact will have versus the transgenic? When you are spraying, actually not even insect, even a lot of other animals die. Say frogs, birds, many things die when you spray insects. But these are much precise. So genetically modified, if you specifically can give insect resistance, one insect, that's much better than spraying insecticide. Organophosphorus insecticides can kill everything, up to human also.
It will affect entire food chain.
Entire food chain, yeah, it will be very specific. This is much better, but for some reason, as I told you, largely could be vote bank. And another way is also there. Don't quote me anywhere. People say that chemical company, fat companies are supporting financially underground to make the anti-J movement. I have come out from here. I am not telling as a teacher here. So many of the chemical companies, because their business will be going down, they are funding underground to the anti-GM group. Just chalo protest karo, because they say $1 billion business, right? Insecticide, pesticide is $1 billion business. So if insecticide resistant crop comes, their business will go down. So they are actually undergroundly paying them for doing the protests. That is also possible. You never know.
So if there is no questions,